All right. Thank you for joining us for the last director's seminar for this semester of academic year 21-22. It's my pleasure today to introduce two of our postdoctoral fellows. But before I do that, I should remind you that our director's search is ongoing. In fact, we have one of our candidates visiting us today, Ravi Alada from Northwestern. He'll be presenting his vision talk tomorrow morning at 10 a.m. in the Beckman Auditorium. So please join us and offer your input. As I mentioned, today's director's seminars will be given by two of our postdocs, postdoctoral fellows, Ryan Hubbard and Autumn Chu. They will each have about half of the session, and I will take and we'll answer as many questions at the end as we can up until about 12:30 or so in the case of Ryan. Ryan Hubbard is a postdoctoral fellow working for Kara Fedemeyer in psychology. He's interested in language and aging and memory. He received his PhD from psychology here at the University of Illinois in 2017, and he began his postdoctoral fellowship in 2018 through 2021. Today he's going to be telling us about probing neural signals of prediction during language and comprehension. Ryan, please join me in welcome.
All right, well, thanks for having me. Can everybody hear me okay? You good on the Zoom? Yeah. All right, so I'm going to be talking about prediction during language comprehension and novel techniques that I've been applying to neural data to try and get this idea of prediction, particularly pre-activation of upcoming information during language input. So if we think about language input, it's pretty rapid and it varies between speakers. So it seems sort of chaotic and fast, and how could we possibly predict anything that's coming up? But on the other hand, language is also somewhat predictable, that there's physical co-occurrences between certain words. There are grammatical rules that we can use to sort of determine whether a noun or a verb or some other type of word is coming up. And additionally, we can use our memory for certain things to know about certain phrases or information that might be upcoming. For example, if you've seen this phrase, the quick brown fox jumps over the lazy dog, you know about halfway through that phrase what's going to be coming up next. So some idioms or phrases that we have some memory representations for, which can also tailor our predictions. Of course, we don't always get it right. Sometimes we finish each other's sandwiches, but for the most part, we can use information about a context to facilitate processing of information. So as a sentence is going along, some information from that sentence is going to sort of tailor our idea of what might be coming up. And the idea is that predictability of upcoming words facilitates language processing. This is shown numerous times through behavioral evidence. So the idea that comprehension is sort of actively generating features of upcoming information. We can see that lexical decisions are faster for predictable words. The lexical decision is basically, is this a word or not? So you show words and non-words, and the predictability of those words influences your ability to sort of rapidly make that decision. We also know this from eye tracking evidence. So if we track people's eyes while they're reading words, we can see that there is less fixation time. You spend less time looking at a particular word, and you actually are more likely to skip predictable words during reading. So if a certain word is very predictable, you might not even really read it. So the idea here is that that's already sort of been activated by your brain, so you don't even really need to read that information. You can just kind of skip over it. Also cool evidence from what are called visual world studies. So these are studies where you are hearing sentences while there are sort of these certain images on the screen. I'm going to try to do the laser pointer here. Yeah, okay. So there are these certain little images here on the screen, like a boy and a cake and a ball. You might hear a sentence about the boy ate the cake. And so clearly there's only one thing that's edible here. And so by the time you hear the word ate, people are already looking, sort of moving their eyes towards the word the cake, because that's the only sort of thing that would be predictable from that information. So I want to take a moment to talk about how we measure predictability, because that's an important part of this. And we'll do a little experiment here. So I'm going to give you a sentence frame where the last word is missing, and I want you to finish this sentence for me. So if I give you the sentence, father carved the turkey with a, how would you guys finish it? Knife. Yeah, so clearly the most common word there that would complete that sentence is the word knife. And since this is such a sort of predictable word, we would call this sentence strong constraint or strongly constraining, because it really constrains you towards that word nice. So I'm going to give you another sentence here. Let's see the famous
Painting.
painting, sure. Yeah, so clearly there's lots of varied responses with this sentence. Actually, the most common completion here is the word actor, and so since this is not so sort of constraining towards one predictable word, we call this a weakly constraining sentence. But these constraint labels are sort of arbitrary, and they're based on a more continuous measure called closed probability. And closed probability is almost sort of like a crowdsourced measure of the predictability of something. So basically what I just did with you, where I give you that sentence frame, we do this with, say, 100 people, and a whole bunch of them are going to say knife. There's going to be one weird person that says cleaver or something, but for the most part, people are going to consistently say knife, and so we can take what is the most modal response of that sentence frame and sort of give the percentage of people that gave that response. In this case, it's very high, so 97% of people will conclude this sentence with the word knife, so that has a closed probability of 97%. Well, we can also say things that are, say, above 50 or 60% more strongly constraining. We can make these sort of categorical labels. And our other example here, we have went to see this famous actor, but maybe singer, maybe painting, again, maybe just person, maybe a clock, maybe a star, etc. Turns out actor is the most common response, but only has a closed probability of 12%. Very few people are giving this response. This is not a very predictable word from the sentence frame. We call this weakly constrained. And sure enough, if we sort of revisit those behavioral and eye tracking studies I mentioned earlier, you see larger facilitation when the context is strongly constraining or when that word has very high closed probability. So we can measure this on a more continuous scale too, and reading sort of fixation times and lexical decision times are related to the closed probability of gender. Of course, we have an issue here where behavioral responses and sort of eye tracking studies can be sort of, people are sort of forced to make a response or may not reflect sort of a standard language processing scenario where you have these very specific pictures up on a screen. And so what we tend to use is actually EEG. We record people's brain activity while they're reading sentences, so we can get at this more sort of natural sentence reading and just measure the brain responses and look at how brain responses vary based on certain characteristics of words or characteristics of sentences, etc. Some of you may already be familiar with EEG and ERPs, or event-related potentials, but the notion here is we give people a whole bunch of trials, and any individual trial may be somewhat noisy, but by averaging over these trials, over multiple presentations of certain words or stimuli, we can find these sort of neural responses that come out through the average, so the noise will sort of wash away, whereas the signals of interest will remain. And we call those signals of interest the sort of event-related potential because it's the amplitude change that is related to the event of interest. And those are going to generally be plotted negative up. Sorry about that. This is just standard convention in ERP literature. But you can see a waveform following some particular stimulus will have certain peaks, and we call those peaks components of the ERP waveform because we'll see amplitude changes in those peaks based on some sort of experimental manipulations or cognitive changes. So the most common ERP we look at in the language literature is probably the N400, named so because it's a negative going waveform that peaks around 400 milliseconds. And while the N400 has been related to all sorts of things, generally we think of the N400 as semantic processing or information or retrieval of features from long-term memory. So if the N400 is very small, it means you didn't have to do a lot of work to retrieve that information from long-term memory. That semantic access was facilitated. And sure enough, while the N400 is not necessarily a measure of prediction, we see that N400 amplitude is modulated by predictability of information and is graded by closed probability. It's kind of what I'm showing you over here. You have smaller N400s for higher closed probability words, more constraining stuff. I'm just showing you that here with our example sentence we came from before. So father carved the turkey with a knife is going to have a very small N400, but if you saw father carved the turkey with a smile, which is kind of creepy also, you'll see a large N400, so that word smile, because it's not as predictable. Again, you see they went to see the famous actor. This is the most predictable response, despite it being a very lower closed probability. So it is a larger N400 than when it is very predictable, but it is still smaller than when it is completely unpredictable. So this is what we talk about, that facilitation based on closed probability. Even with this electrophysiological data, though, there's kind of an issue, which is that when we say, father carved the turkey, we get the word knife. and we see some different in our N400 response. What we're sort of doing is we're looking at a response after the word and inferring what went on prior to that word. We infer that people are sort of predicting this thing, and that leads to this change in the N400 response. But our idea of prediction is really tied to this notion of pre-activation, right? You've pre-activated these features in the brain, and that's why that semantic axis is easier for the N400 because it was already kind of active to begin with. But we don't know what's going on in this sort of pre-word activity, so we want to try and get at this using a different measure. So the idea is these studies generally focus on consequences of prediction, but we want to see prediction-related activity before the stimulus. We want to see this pre-activation in the brain. That's what I was trying to get at the start of my postdoc, using a technique called representational similarity analysis. We won't go through everything, but it's generally a method that was originally designed for functional magnetic resonance imaging. The basic idea is that when you see a stimulus, you'll get some pattern of brain activity, which we can call sort of a representation of that stimulus, and we can relate representations of different stimuli by sort of correlating these patterns of brain activity. Well, this was generally done with fMRI and sort of a spatial location. What is the hippocampus doing? What is the visual cortex doing? We can generalize this to EEG by using the spatial pattern over the scalp as our sort of representation of the stimulus and see how this representation unfolds over time. So the notion here is we have father carved the turkey with our knife, our favorite sentence at this point, and we're going to call this the pre-final word, and we're going to call this the final word where we know what the close probability of that word is. And so if we look at what's going on when you see the pre-final word, you have some channels, EEG channels over the scalp that have activity following the pre-final word, and you have the same thing following the final word, some channel activity across the scalp following that final word. And so all we're going to do our RSA is take the vector of channel activity at time 1 following the pre-final word and the vector of channel activity following the final word and correlate those two vectors of channel activity. And then we're just going to basically do that for time point 2, time point 3, basically where you're sliding across gives the correlation at each time point. And what this does is it gives us a correlation time course of those patterns of activity across the scale. Why do we want to do this? Well, the notion is if the representation of the final word is already active prior to its appearance during the time of the pre-final word, the similarity between the pre-final word activity and the final word activity will be higher when that close probability is really high. So we should be able to see some sort of graded pattern of neural similarity based on the close probability of that final word. This will give us an idea that that final word is already activated at the time of the pre-final word. Let me just keep going here. My animations got screwed up here. So if we look at the similarity time course, time gets a little confusing here because time point 2 would be 200 milliseconds after the pre-final word and 200 milliseconds after the final word, similarity between those two patterns of activity, right? And what we see is there's an early peak in this similarity, much earlier than our N400 response. And if we break this out by condition, we see the greatest similarity for those words that are strongly constrained and expected. So namely, those words with very high close probability, you see a very high neural similarity between the pre-final and final word, and that actually is lower when the constraint is lower than close probability. Of course, again, these constraint labels are kind of arbitrary. What we can also do is take this measurement, take a sort of mean similarity measurement within this time window and relate this on an individual word basis to close probability on a more continuous scale. And what we see is that neural similarity is indeed sort of linearly graded with the close probability of that word. The notion here is that we have some evidence of pre-activation in the brain. The pattern similarity seems very tied to the close probability of that word, suggesting that you're sort of pre-activating it more when it's more predictable. We've also looked at this across different words. So if we were looking at sort of final to pre-final, we get this very strong effect. If you go final to sort of pre-pre-final to see if this sort of pre-activation is already occurring 2 words back, you can see this is already starting to fall apart. The effect is not very strong here. And here, if we go 3 words back, there's nothing at all. There's no difference between our conditions here. So the notion here is that there's evidence of pre-activation, and it seems very linked to sort of this rapid timing, maybe sort of more automatic association mechanism, where when you get that pre-final word, it activates features of the final word. So that was published in 2021. We are now using this method to extend to new domains. So we're, like I just said, I'm very interested in aging. Part of the interesting thing that we see with older adults when they're reading is they don't seem to show the same patterns of N400 responses relating to closed probability as younger adult readers do. I apologize, my animations got screwed up, so there should be text too. But the notion here is if we take our strongly unexpected word and compare that, the N400, to a strongly expected word, you'll see a large difference, as I showed you before in that N400 plot. But if we look at that with older adults, that difference is much smaller. We don't see the same sort of facilitation based on closed probability. Right? So one of the arguments here is that older adults don't seem to be engaging the same predictive mechanisms as younger. However, remember what I told you before, that we're sort of inferring this pre-activation, this predictive mechanism based on a signal after the word, right? So we can use the same RSA method to look at the pre-activation in older adults compared to younger adults and see what happens there. Because we do have some evidence that, well, maybe prediction is preserved in older adults. So this was a study by Trevor Brothers' group, or Trudy Dave, 2018. So if you just give people explicit instructions to predict a word that is coming up, and then you ask them, was this the word that you predicted? You can really compare words that were predicted and it matched, and words that were sort of not predicted and it didn't match. And you could see this difference in an N400 that is sort of actually more timing-based. So that you get this prediction match effect that's earlier than this more general context effect. And while you can see this N400 of sort of amplitude is generally smaller with older adults, you still get this separation of a prediction effect and a context effect in the older adult readers, suggesting that maybe there is some preservation of prediction in older adults, and so to say sort of, well, they don't predict at all, might not make as much sense. So if we look at older adult readers reading those same materials, father carved the turkey with a knife, and we do the same RSA analysis to get this idea of pre-activation in older adults, what we see is that while the peak is a little bit weirder and noisier, we see the same effect where strongly predicted words have a much higher similarity than more weakly expected words. And again, if we take this and measure, on a single word measure related to closed probability, we see a linear relationship between that pattern similarity and that closed probability. So while those signals may be a little bit smaller or more reduced in older adults, it does seem like older adults are maybe engaging in the same pre-activation mechanism as older adults, sort of write off their prediction skills. Maybe not be the correct way to go about this. Got to wrap things up. Tell you about one last study we've been working on. So I've been telling you about the closed probability of the final word and how a sentence constrains during that final word. But we're also interested in how prediction is engaged across the sentence. So do we see the same prediction mechanisms engaged across an entire sentence? So we took a whole huge group of online individuals and had them do a closed norming task, but for across the entire sentence. So instead of getting father carved the turkey with you get what the final word is, you get father, and you have them try and guess what the next word is. Then you get father carved, and you try to guess what the next word is. We have them do this throughout the entire sentence. We have hundreds of people do this, so we get a large sense of closed probability across an entire sentence. And so we can get closed probability of every word of every sentence, as well as average reaction time to type that word. So we get a sort of behavioral measure that we can relate to predictability. And since these specific sentences have been used in numerous EEG experiments in the Federmeyer lab, we just gather up all of that EEG data using those sentences and relate it to our closed probability across the sentence. So we have a huge data set where we can explore prediction on a word-by-word basis across the entire sentence. And what we see in the analysis we've been doing is that across the sentence, this neural similarity measure, if we get the neural similarity between this word and this word, this word and this word going across the entire sentence, is related to how predictable that word is across the sentence. You can see it's kind of noisy, but again, it's a huge data set, so this still comes out significant. So there's a relationship between predictability and neural similarity across every point of a sentence. And if we look at how neural signals relate to reaction time to respond to input words during the closed probability task, or the closed, we see that neural similarity predicts reaction time, and again, in a negative line, which you would expect. You're faster to produce words that have more neural similarity. And interestingly, we find that N400 amplitude doesn't predict response time. So this, signal that's generally thought to be more related to closed probability doesn't seem as behaviorally related. We may have sort of a dissociation here where the rapid pre-activation signal might be more related to reading and sort of behavioral responses, whereas N400 might more reflect this semantic memory access, the sort of more higher cognitive process. Right? All right, so I'm going to wrap things up here because I don't want to go over here. But we have the first evidence of rapid pre-activation that's sort of graded by close probability during language comprehension. It seems very tied to this pre-final word. It's something that's sort of yoked and maybe sort of this automatic association process. And older adults seem to show similar pre-activation, though it seems maybe a little more reduced compared to younger adults. And what we're seeing is that predictability influences neural signals across words of a sentence. It doesn't seem just tied to the final word phenomenon. And this relationship between similarity and close response times suggests a behavioral relevance of those neural signals. So we're thinking about how this relates to other work in the field on predictive coding. Predictive coding is this notion of a more general brain mechanism, sort of hierarchical organization, where you have top-down neural systems they're sort of communicating with sort of sensory processing cortical areas to generate predictions about upcoming information. We see this in sort of primates and with these more lower level, very, beeps and boops and small sensory responses, but this may scale up even to the higher order world of language, where these predictive mechanisms are still at play to predict upcoming information. We have open questions about how this rapid pre-activation might be observed during more natural reading, if we can relate sort of eye tracking measures of people just reading without this sort of timed response of the the way we run these experiments in the lab. And are these signals related to reading behavior? Do you also if you're more likely to skip a word, do you also have stronger pre-activation? And then are these individual differences that relate to engagement of these predictive mechanisms, and are those individuals consistent across age? For example, if you have more executive processing skills or rare visual acuity, there's all sorts of things we can look at with relationships to this pre-activation. And finally, on a more neuroscientific level, what are the neural generators of these predictive mechanisms, and do they overlap with these more domain general signals that we see in primates or with this predictive coding framework? Can we relate these two things together, the sort of low-level brain hierarchy mechanism to this high-level reading mechanism? All right, I'll wrap up there. Just thanks to Kara and everybody in the Cognition and Brain Lab. Thanks to the funding folks that allow me to do work, and thanks everybody for listening. I hope everybody heard that over them.
We'll take questions, yes. Are there any on?
Question.
Yeah.
Do you see some of the same effects if it's not language, but if it's like visual stimuli, like you're watching a scene of a movie and something unexpected happens versus the standard thing you expect to happen?
Yeah, there's some interesting work on that, especially we've got Peter Hawk, I think, who has done some cool studies where you have more of these low-level visual stimuli that are predictive or not predictive. He's done some cool things where you sort of have a train of visual stimuli, and then on some trials, the train of visual stimuli starts and then it ends halfway. And so what the idea is, do you still see pre-activation of those ones that didn't occur? And it turns out you do. So you can still sort of see pre-activation of things that don't even actually happen.
What do you consider an older adult, and do I also do any studies with kids?
So an older adult, I believe our cutoff is 65, so we're doing like 65 to 80 around there. We in the Federmeyer lab have not done any work with kids, so we're looking at the older side of the spectrum. But it'd be interesting to see if there's a certain age where these predictive mechanisms develop, if we can sort of relate that to general literacy skills in the future, those sorts of things. So that's definitely an interesting question.
Your studies were.
One question on the Zoom.
Oh, yeah.
Might the advantage of neural similarity over N400 in predicting reaction time be due to just having more data on brain activation?
More data on brain activation? But not sure I entirely follow that question. I guess, I mean, the pattern similarity measure is actually a little more noisy and sensitive to fluctuations. So if you have like larger alpha oscillations, that pattern similarity can be affected by that. And whereas the N400 is a larger time window mean signal, so I think actually the N400 responses tend to be a little more stable than the prediction responses. More brain data is helping us much more with capturing these predictive responses than the N400. The N400 is a little more robust in those kinds of things, I would say. I don't know if that answers that question.
Can I follow up?
Yes, go ahead.
Oh, sorry. Yeah, so I guess what is that, so the N400 is just a very particular time window, and I'm wondering if, given that you've got this, you're collecting electrical data from all over the brain, right? I mean, that just seems like a, even though it's, I mean, I get it's a new measure and it's a lot of noise to it, but I think it's just a fascinating measure. And I wonder if it's just that you're capturing, more information temporally and spatially. So that gives you a little bit more of a signal, a different kind of signal, but maybe more of a signal.
Yeah, I think I get what you're saying. So there's been, one of the influences to do this study was the interesting studies with, I'm trying to remember, it was visual stimuli that sort of differed in visual angle in very small ways. And these sort of pattern classification or pattern similarity approaches are able to identify these very subtle changes in visual angle, suggesting that there is, information that can be captured that subtly changes in scout patterns, which is why we wanted to do this at like sort of this word level, right? Of like, maybe there is some word level representation that's sort of captured in some scout pattern. We can see that recapitulated or capitulated, I guess, prior to the word itself, and we can capture that spatial pattern. So I agree, it's a different signal than the N400 for sure.
Yeah, thank you.
So would you expect that the prediction for people from different cultural backgrounds or economic backgrounds are different?
There's been some work in that in the lab. Actually, Chicago has done some work on that, looking at sort of literacy and socioeconomic status and how those things relate. I actually don't know if the pre-activation signal would be affected, because we see those things at the level of the N400, where maybe it's just more semantic memory access and how your semantic network is sort of defined, whereas this might be more of an automatic associative, boom, this word goes to this word, which may not be as affected by these types of things, right? So, like we saw in the older adults, we still see some evidence of this pre-activation signal. So it could be the case that, these pre-activation signals aren't as affected as much by those sorts of things.
How is the pre-activation thing produced in an older adult? Like, don't they have more experience?
Yeah.
And so, shouldn't they be more able to predict those things before they even occur? So,
it's a good question. One thing we're definitely considering right now is how much our amplitude, the sort of raw overall similarity signal you get, is influenced by more cognitive things versus influenced by more just physiological things, right? So older adults generally show lower amplitude responses to various things, which may just be due to, aging leads to a different neural response, right? So if your neural response is maybe a little noisier or maybe a little smaller in general, the correlations that you're going to get between those patterns might be a little noisier, might be a little smaller. But our point with that study was, if you look at it on this, single word and relate that to closed probability, that relationship is still very strong. So while the overall similarity might be reduced in neural adults, it might not be meaningful. It might just be something that's physiological.
Well, maybe we have two questions if they're quick.
So it was kind of a follow-up question to that. What about an older adult learning a new language? Would the familiarity with, a different language influence the amplitude of the informant, for example?
Yeah, that's a great question. One I'm not as well versed on. There's definitely lots of interesting work on bilingualism and second language learning and how N400s relate to that. I have no idea how the pre-activation signal would change based on your second language or your familiarity with a second language. That's definitely an interesting question. So.
I noticed that your closed probability x-axis had kind of a 0 to results in the middle, around like 75%, and 75. What was the actual distribution of the probabilities? In
so we leave, in those experiments, the sort of 45 to 60 range or something like that is actually left out, because there's interesting phenomena that occur when you have sort of like a 50-50 split with those kind of things, because you have different neural responses entirely, where you get these maybe sort of like reconsideration responses, we're kind of calling them, where you're like, oh, that's not what I was thinking of, but that totally makes sense. And you get these like larger frontal things. So those studies actually tend to kind of leave those things out, which has pros of not sort of having to deal with those neural signals, but cons of we are missing a chunk of the closed probability. But there are a couple studies which I'm planning on analyzing, we've done in lab, where we have the full range of close probability, and I'll be looking at RSA signals across that full range.
Thank you.
Yeah.
So please join me in thanking Ryan for a great stimulating work. Autumn, are you all set here? Let me take a moment to get Autumn set up, and then I'll introduce her. Autumn is a postdoctoral fellow with Jonathan Sweedler in chemistry. She earned her degree from the University of Minnesota in 2018. It was during the period of 2018 to 2021 that she was a postdoc fellow here at the Beckman Institute. Autumn is about to begin a new position at Michigan State as a assistant professor in the chemistry department. Today she's going to talk about her work that she did with Jonathan and here at Beckman, de-alanine in the microbiome gut-brain axis, sources, biodistribution, and functional roles.
Thank you, Jeff, for this very kind introduction. So, yeah, I'm going to talk about a different kind of language, which is the chemical language, the molecules. So, de-alanine in the microbiome gut-brain axis, and I should mention that this work is made, like, big part of this work is made possible by the support of the Beckman Fellowship Program. So, I feel extremely lucky to be part of it. Okay, all right, let's get to it. So, the microbiome gut-brain axis. In the Beckman, a lot of us care about the brain, the central nervous system. And you might also have heard about the saying that the gut is our second brain, which refers to the enteric nervous system. So the enteric nervous system has similar chemical foundations, just like the central nervous system. And the crosstalk between the gut and the brain has been shown to have a lot of importance in different diseases, including the neurological diseases. And on the top of that, there's the microbiome, which is a lovely diversity, a very diverse collection of the microorganisms, trillions of them living mostly in our gut, called the gut microbiota. So those gut microbiota, they are very diverse. They have their own activities. They make a lot of molecules, they metabolize a lot of biomolecules that they use to communicate with the host, which is animals, all the time. And that's what we call this, and what we care about, the molecular basis of this microbiome gut-brain axis. So why is it important? The altered microbiota has been shown to correlate to many diseases, including neurological diseases, the irritable bowel syndrome, central nervous system diseases like depression and autism. So currently, generally in the field, there are generally thought there are 3 different pathways that the gut microbiota can communicate with the nervous system. So there is the HPA axis, the hypothalamus pituitary adrenal axis, that involves a lot of immuno responses and endocrine pathways. The gut microbiota can also potentially directly communicate with the nervous system through the vagus nerve. And then the third is about the alteration of the neurotransmitter level, so the chemicals that our nervous system use. So the microbiota can either directly, or they can both directly produce different neurotransmitters, or they can alter the host production of the different neurotransmitters. So among all these different mechanisms, I'm mostly interested in the direct production of the neuroactive chemicals, the neurochemicals. If you compare the metabolic profile from the gut content in the regular mice or conventional mice that has a healthy microbiota to the gut content of the germ-free mice, which means that they have not even a single bug within them detectable, you see that the neurotransmitter levels in the conventional mice are much higher than the germ-free mice. And then the microbiota has a huge genetic pool, and a lot of them resembles the biosynthetic pathways of these neurotransmitters. Moreover, correlations were found between the enrichment of the neurotransmitter genes and the samples, gut content samples from the autism patients. So there are possible links being revealed pretty much every day. This is a really rapid advancing field about how the production of the neuroactive chemicals and other activities of the gut microbiota might regulate the neurological diseases in the human and in the animals. Right, so that was the general background. And then the specific set of molecules that I was interested in, or I am still interested in my postdoc research, is about the amino acids. So the amino acids, they are the enantiomers, the mirror images of the prevalent L amino acids that we're all very familiar with. The L amino acids, they are found in for the building blocks for the protein synthesis, energy sources, precursors for a lot of different metabolites. But how about the amino acids? So long time before, the analytical technique was not that advanced, and people used to thought that there's no amino acid existing in the higher animals. But with the advancement of the analytical techniques, we found researchers actually found that this very small level or small amount of the amino acids existing in the animals and higher animals, and some of them have very important physiological roles. So one of the most important roles of the amino acids are the D-serine as an endogenous neurotransmitter. So the D-serine can bind to the NMDA, the glycine side of the NMDA receptor. This is the N-methyl D-aspartate receptor. The D-aspartate can bind to the glutamate side of this NMDA receptor. So the binding of both sides together with the membrane depolarization can activate these NMDA receptor, important for what we call the long-term potentiation. And the long-term potentiation is a very important activity of the neurons for the synaptic plasticity and the formation of the memory. So there is actually a lot of studies showing that the D-serine itself and also the enzymes that are involved with synthesizing and metabolizing the D-serine in the neurons and the astrocytes, so in the nervous system, are related to neurological diseases and can be used as a therapeutic for the different disease treatments. Right. So what's interesting about the D-alanine? So the D-alanine, as you see, that the structure is very similar to the D-serine as I'm shown here, and you expect it might also bind to the NMD receptor. And that has been shown that the D-alanine also is a very potent coagonist that can bind to this NMDA receptor in the in vitro experiments. It is also found in many parts of the rodent of the human body, including the serum, the brain, and endocrine glands, those that may perform important neuronal and endocrine functions. The interesting, very fun part of the D-alanine is that there's no endogenous synthesis pathways that is found in the animals. So the D-serine has the serine racemase to synthesize the D-serine from L-serine in the neurons, in the astrocytes, and potentially in some of the endocrine cells as well, but not the D-alanine. On the contrary, the D-alanine was thought to be mostly originated from the microbiota. So the bacteria has what's called alanine racemase that converts the L-alanine to the D-alanine, which is a very essential component of the bacterial cell walls. So we have a hypothesis here that the D-alanine could be a potential signaling molecule for this host-to-microbe interactions, while still there's a lot of things that is unknown about this molecule, and that's what I was interested in. So while I said that the in vivo function of this molecule is not very clear yet, there is some relevance has been shown from the previous studies. For example, against the activation of the NMD receptor in vitro. And very interestingly, the D-alanine was found to be colocalized in the insulin-secreting beta cells and the ACTH-secreting cells in the pituitary gland. So that potentially points to the involvement of this molecule in the glucose metabolism. This is also something very interesting, that there is a day-night variation shown about the level of the D-alanine in the plasma, pituitary, and pancreas in the mice. And finally, it was found to be correlated to the disease status, such as Alzheimer's disease, diabetes, and also has been tried as therapeutic add-on for the schizophrenia in both animal and actually preclinical studies here. Right. So the one key to understand the potential role of this D-alanine is to determine its origin in the higher animals. So we say that there are three potential sources of the D-alanine in the mammals. One is the diet, and one is the microbiota. And I put a question mark here is about the endogenous synthesis. So by comparing, this is a previous study a while ago, comparing the levels of the D-alanine in the specific pathogen-free mice, so mice with healthy microbiota, in the germ-free mice, and they show that there's a huge difference in the D-alanine levels in these two mice. So that points to the very possibility of the microbiota either contributing to the D-alanine level in the animals or changing the D-alanine levels in the body. Another very interesting study showed that there's actually much higher level of the D-alanine in the saliva samples compared to other tissues. And they did a conversion experiment that showing that the saliva samples can convert the L-alanine to the D enantiomers, but it cannot rule out the activity of the oral microbiome. So endogenous synthesis is still a potential pathway to be a source of the D-alanine in the mammals. The first thing we were interested in to see is that can the gut microbiota actually produce the D-alanine? So this is one of the very first beginning collaborative work when I get started here. The gut microbial species, so collaboration with Hui Mingzhao's group in the chemical engineering. So the gut microbial species from the rat feces were cultured on the plates. And then after culturing, the biomasses were collected. And we used a different measurements, including one, the enzymatic assay that is specific for D-alanine and other neutral D-amino acids, and also the mass spectrometry assay to measure the amount of the D-alanine and other D-amino acids produced from these gut microbial species. So we're pretty sure that these gut microbial species can produce a different D-alanine. And it's very interesting that you see that these are all the different, the same species, but the different strains in the same species, but they can actually produce quite a different amount of the D-alanine. So that also shows that there is intra-species variation that you won't be able to catch using the sequencing, but you have to do some chemical profiling to catch those variations. The next question to be answered is that, okay, now the gut microbes, they can produce the D-alanine. And can this D-alanine be absorbed through the gut? And if so, what is the biodistribution of these gut absorbed D-alanine? And the other thing is that another very important question is that, are there endogenous alanine racemase activities in the animals to convert the L-alanine to the D-alanine? So think about the conventional mice model that it has a microbiota that has a lot of activities regarding the synthesis and metabolism of the D-alanine. And that is an interference for us to understanding these questions. So I fully utilized the newly built nonobiotic facility on the campus. I used the germ-free mice. So by using the germ-free mice, we exclude the interference from the gut microbiota, and we can then use the stable isotopically labeled L or D-alanine to see the conversion and see the biodistribution through the gut. So this is the feeding experimental setup. I'm just going to quickly go through it. We have two different isotopically feeding experiments. One is a forced feeding of the gavage, directly deliver it to the stomach, and it's a high dose. And the other thing is, and then we sacrifice that after 1 hour. The longer treatment is adding that a low dose into the drinking water and let them drink for about 2 weeks to see the long-term accumulation of these molecules in the mice tissues. And then we did the tissue collection, including brain, pituitary gland, excuse me, the salivary glands and islets and acinar tissues, and some gut tissues, the blood, and the gut content as well. So, and these samples went through the amino acid extraction, cleanup, the zeroitization, so that we can separate the chiral L and Z amino acids on the liquid chromatography. And then we use the mass spectrometry to do the quantification of these different molecules. Okay. So this piece of the result is, this is control mice, but not related to the isotopic as well, but we can compare the different amount of the D-alanine in the germ-free mice and the regular mice. So not really surprising that the colon content, which is where most of the microbiota is, has the highest amount of the D-alanine. And there are differences that are higher levels of the D-alanine in some tissues in the regular mice compared to the germ-free mice. So something interesting is that the D-alanine is not very different between the germ-free and regular mice in tissues like the brain and the small intestine, and also acinar and islet tissues. So the question may be like, do they make their own D-alanine that is not affected by the microbiome, or do they just sort of maintain a D-alanine homeostasis themselves? So that's the question that we can try to answer with the isotope tracking experiment. Okay. Oh, last piece of results, mouse chow actually contains a lot of the D-alanine, so it's likely the diet to be a source of the D-alanine for these germ-free mice here. Okay. So one of the important pieces is about how the orally administered D-alanine is absorbed and distributed from the gut and to the different tissues. So each data point here is the level of the D-alanine that is normalized to the total unlabeled alanine in the same sample, and also the D-isotope level in the plasma of the same animal, so that we can do cross-tissue and cross-treatment comparisons. So here, this line means that it's normalized at the plasma level, and you see in most of the cases, the plasma has the highest level of the D-alanine in a lot of the tissues and the bio samples. So the first thing we notice is that in both settings, the acinar tissues and islets in the pancreata, they have similar levels of exogenous D-alanine, just like similar to the plasma. And the next very interesting thing is the difference between the two oral administrations in the brain and the pituitary. So you see that in the short term, which is the gavage, we only waited for 1 hour. The brain has very little amount of the uptake of these gut-absorbed D-alanine, as similar as the pituitary. But then after feeding them for 2 weeks, somehow these D-alanine from the gut can accumulate in the brain and the pituitary. So that means that these D-alanine through the gut can get across the blood-brain barrier, and the brain somehow has a mechanism to store and accumulate these molecules in their tissues. And the last thing is that it seems like both the colon and the small intestine can absorb and retain some part of the D-alanine from the oral administration. So the transporters for these D-alanine in the gut is still not clear yet. So this might indicate or point out some directions to look for the transporters in the different gut tissues. So next question that we can answer with the germ-free mice, again without interference of the microbiota, is that is there endogenous synthesis activities from the L-alanine to the D-alanine in the mouse? So if there is, or if there were alanine rate misactivities, you will see this is through this carbanionic intermediate, which means that if you feed this with the L-alanine 2,3,3-D4, so alanine plus 4 in the mass, you will see the anticipated product of the D-alanine 3,3-D3. So that means that it's different retention time on the liquid chromatography and also different M over Z in the mass spectrometry that for us to detect and quantify. So this is the result of the LED conversion in the germ-free mice. So you can see that these are really not different from each other if you compare the dosed group and the controlled group, and most of them are very close to the background level. However, something very interesting happened here is that you might see a difference in the salivary gland here. So I was a bit excited about that. Maybe that means that the salivary gland has some activities for this conversion. But coming back and checking the original spectrum, I realized that they are all under what we call the limits of quantification. That means the quantification was not as reliable. So these are still mostly background level spectrum. So to make a comparison, what if we feed those we feed those isotopes to mice with the healthy microbiota? Well, this is what happens only after 1 hour, and you see huge amount of the alanine conversion product distributed into different kind of tissues only in 1 hour. So we can conclude here is that really a lot of conversion activities happen in the microbiota instead of in the tissues of the mice themselves. So that's the result from this by distribution and conversion assays. We found, interestingly, that these gut-absorbed D-alanine can accumulate in the brain and pituitary over time, always the highest in the acinar and islet tissues in the pancreata, and it can be absorbed from both small intestine and colon. And we conclude that no alanine rate activity can be reliably detected in the germ-free mice, but very quickly detected in the regular mice. The next question that or the other question that I was very interested in is that, okay, so what can we look at the potential function of these small molecules in vivo? Germ-free mice is a great model, but it's very expensive and it's kind of hard to work with. It costs a lot of money as well. I want to I would like to have another animal model to study, to explore the physiological roles in a preliminary way. So the question is that if the microbiota change the D-alanine synthesis and metabolism, how will that affect the animals? So I used this C. elegans, the models. It's the cute little worms that is very widely used in the neurobiology area. It has a lot of advantages, particularly to connect the genetics to the phenotypes. it has a quick life cycle, so only 72 hours, then you can have the adult worms to measure on, to do your measurement on. A lot of advantages, phenotype characterization, a lot of things that you can look at regarding the animal physiology, and actually very easy microbiota manipulation make them ideal for the microbiome hosted studies, because these worms, they actually are fed on the bacterial diet. So to make the worms associated with a specific microbial background, you just feed them with a different kind of the bacteria, and they will start to colonize the gut. They have very tractable host genetics, again, to connect the genetics to the phenotypes. Okay. Are the D amino acids relevant for the C. elegans? And the answer is yes. So first, the C. elegans also has an NMDA receptor. It's a pretty conserved one. And I detected the D-serine and the D-alanine in the C. elegans, as well as their bacterial food and the nutrients. So actually from this here, you can see that it is very likely that the D-serine is mostly to be originated from the endogenous synthesis, while the D-alanine is very likely to be the exogenous, so similar to the mice studies. And the NMDAR is related to C. elegans locomotion, was shown earlier, that it's, so you can use the C. elegans locomotion to kind of infer to the NMDAR activities. Okay, so now the question, when we fixed our model organism, another question becomes, if we change the microbial and the alanine synthesis and the metabolism, can that affect the C. elegans measurable phenotypes? So to do that, first, I found that there are two different alanine race misses that is responsible of making the alanine in the microbes in the E. coli. So they're alanine race miss 1 and 2. And another very important another very important pathway is the D-alanine ligase that is use the D-alanine and for the bacterial cell wall synthesis in the bacteria. So I found the single gene knockout mutants of the E. coli from the KO collection. So these are the mutants that has the single gene knockout. And what I did is that I fed these mutants to the C. elegans and also challenged them with the high glucose or not, if you remember that one of our hypotheses is related to the glucose metabolism. And I perform a lot of phenotype analysis on these worms that are fed on different bacteria mutants. So I have a lot of results here. Due to time, I want to only show a couple of very interesting ones. So the first thing is that I found that the E. coli D-alanine metabolism can affect the worm swimming behavior, including the worm initiation rate. So that's a part of the swimming activity. That the D-alanine ligase knockout, this one, can result in a decreased amount of the swimming activity of the worms. And then also the alanine race miss 1 knockout can increase the body amplitude. So these are all the parameters of the C. elegans moving in the liquid medium, like how frequently they're bending or how deep they're bending their bodies. So those are involved in for example, the activities of the motor neurons. And I hope this result in the future can be more extrapolated to the activities such as the gut motility in the animals, in the higher animals. The other very interesting thing that I found was actually kind of unexpected, but that's part of the beautiful in the science, is that I found that the interaction between the DDLA, bacterial DDLA activity, the high glucose, and also the C. elegans lifespan. So the lifespan is that you monitor a population of the animals and see how long each one lives, and you plot the survival plot. So under no glucose conditions, it seems like these different bacterial diets does not affect their lifespan. But when you challenge them with the high glucose conditions, and the diet of the DDLA mutant, which is the D-alanine ligase, significantly reduced the lifespan. So there is an interaction of the reduced lifespan, the high glucose, and it only happens when you feed it with this bacterial mutant strain. So I want to understand a little bit more about what happened here. Maybe it's because the C. elegans has some sort of preference of the different food that affects their lifespan. So I checked that there's food preferences, and long story short, that there's a lot of data here, but is that the C. elegans larvae, it can avoid certain bacteria, but under certain conditions, like how old they are you on the no glucose plates or the high glucose plates? But that does not seem to correlate to the lifespan reduction. So this lifespan reduction, again, I want to look at the mechanisms. It seems to be mediated by the bacteria, the D-alanine ligase activity. So this activity or this enzyme is very important for the assembly of the peptidoglycan layers of the bacterial cell walls. And looking at the literature, I saw that the bacterial peptidoglycans, they can be recognized by the host innate immunity through the receptors. So a lot of these receptors are very conserved, including the toll-like receptors or these MAPK signaling pathways. So the experiment I did is that I challenged them again with the high glucose, but instead of using wild-type C. elegans, I used the mutant C. elegans, which has a knockout of these genes, either the TOL1 or the PMK1. So we're not seeing the reduction in this case in these mutants. That indicates that these pathways in the C. elegans are probably involved in this bacterial DDLA-mediated lifespan reduction. Okay, I think I'm approaching the end. So these are the conclusions that I got for this very interesting small molecule, the D-alanine. It has there's biodistribution through the gut in the animals. The different sources, it's very likely from the diet and the microbiota, but not from the endogenous synthesis in the mice. And using the C. elegans, I explored its potential physiological functions under different conditions and find the correlations between the different phenotypes, the bacterial diet or the bacterial enzymatic activity, and the high glucose condition. So I come to this technology. So these are all of the people that I worked with in the 3 different labs. The Sweetler lab and Schroeder lab, and also the Huining Zhao lab. And shout out to the rodent nodal biotic facility. That is really, all of them are very, very helpful in setting up the germ-free mice experiment, the flow cytometry core, and also the Beckman microscope suite, and all of the funding sources, including the Beckman Postdoc Fellowship. And yes, this is where I am going to. If you're interested, please keep an eye on my future work. So I was very lucky to be trained here in this interdisciplinary Beckman Institution, and I plan on keep this spirit going on in my future lab. So my lab in the plan, it might change, but in the plan, it's going to combine the analytical chemistry, the toxicology, which was my PhD background, and also the neuroscience, and look at this microbiome gut-brain axis on the different environmental exposure. So thank you very much for your attention.
Thank you, Autumn, for a very informative and quite a lot of work there in a short period of time. I will start with a question. Yes. So did you ever find the origin of the day-night differences? What does that arise from?
Yes, so I never looked at circadian rhythm. I would love to, particularly with Martha here, but I did not find the bandwidth for that. But that study that I showed, they thought it's very likely to be correlated to the differential gut absorption of the D-alanine from the gut content. So they tested the levels of the D-alanine in the gut tissues and found that the differential amount day-night as well. So they thought it's very likely to be the gut differential absorption.
How are they absorbed? How are the amino acids absorbed? Is there a transporter for them, or do they just piggyback on sort of amino acid transporters? Or?
Yeah, that's a great question. So there are different amino acid transporters, and so far as I know, there is not a transporter that is very specific for the amino acid. For example, like the NMDA receptor, they are very specific for the D-serine and the D-alanine, not the L1. But that seems not to be the case so far for the amino acid transporters. So they may be just like you said, just to go through a general amino acid transporters. But again, there are different kinds of amino transporters in different tissues, like in the small intestine and the colon. So that's also another very interesting thing to explore in the future as well.
Any online questions? All right. Well, thank you very much, both Ryan and Autumn, for this. Will you be watching for papers for the landscape?
Yes, thank you very much.
All right. Thanks for joining us, everyone.
Bye,
Online
everyone.
too.